Ged, Dr. Brady, thank you very much indeed for giving us a few minutes on e-cancer. You're the genomics group in the Paterson lab, which is part of the Christie complex in Manchester.
That's correct, yes.
Now, you're at the European platform meeting here in Berlin. What do you think of this idea of institutes working together?
I think it's superb, actually, because it gives you a great chance to create and normalize cancer therapies and biomarkers, which is a large part of what we're interested in at the Christie and the Paterson, and demonstrate that you've got something that's of potential benefit to patients. So it's an ideal way of learning. I think one of the things, I'm new to the area, and Nick Jones set this up with the Paterson originally, and I've stepped in as the representative. And I found it an ideal, or a very good conduit, if you like, to learning about what's going on in other cancer centres throughout Europe, and starting to share methodologies and results with them so that we can both benefit, or all of us benefit from that. I mean,
as the layman, I would have expected you to do this anyway. So why don't institutes work together? Why did we need to get some EU money to put all these wonderful teams together?
They do, but in practice, the way we work together is through journals and publications and conferences. I mean, effectively that, because that's the easiest way to learn what's going on elsewhere outside of your laboratory. But in practice, you have to have a time, effectively. So what we've done here with the two meetings I've been to for the Eurocan, we've just set aside time where you're in the room with the same people who are doing similar work to yourself, and you have the opportunity to learn what they're doing and talk to them and make those connections. So it's simply a matter of I've heard it talked about as glue, and I think that's a fairly good analogy, because it allows you to sort of bring together things that we're all aware of it, roughly what's going on, and we see a lot of the same people at different conferences. But because it's a dedicated platform with bringing together everybody as the aim of the platform, it tends to be more focused, and you get more direct collaborations emerging from it.
So what's the Paterson bringing to the party? What are your key platforms that you're sharing with other people?
The Paterson itself is the research arm of the Christie Hospital, and the Christie has now set up the largest phase 1 oncology trials unit in Europe. So there's a big input there in terms of therapeutic strategies with Carolyn Dive and Malcolm Ransom connecting with the people in the program. The other thing, as I've already mentioned, we have a very strong theme within the clinical and experimental pharmacology group, which I'm part of, in developing and validating biomarkers. So we're very much interested in finding out what pieces of information we can get from a patient sample, typically blood, that will allow us to improve on therapy selection and get information about what will happen to that patient. So we're one of the forerunners in that area, in the area of circulating tumour cells and circulating free DNA, which are tumour components you can get out of a simple blood sample, and we're working with the other groups in the Eurocan platform to see if we can pool our resources and come up with improved strategies and biomarkers.
And which platforms have you found amongst the other institutes which you're actually now, thanks to this glue, able to exploit?
Well, yeah, we've talked to the Italian groups around about the CTC technologies, and they're aware of in Milan. So in fact, we're collaborating with other groups there already outside of the Eurocan platform. So I'm actively collaborating on groups there to look at cancer stem cells. But in the specific context of the Eurocan platform, we're giving advice and receiving advice back about the different methodologies and platforms you can use for CTC isolation. So the circulating tumour cells are very important, I believe. You can gain a lot of information from them. The technologies that are used to isolate them and enumerate them are varied, and they are either based on cell surface epitopes, which allow you to enrich for the circulating tumour cells, or physical properties like size. And you can have platforms based on both of those, or either one alone. And we're evaluating both of those types of platform, and I've given advice on that to the group in Milan, and they're now trying out the same platforms. Okay. In a way, the CTC platforms complement one another. The Veridex system, which is the magnetic bead antibody system, is FDA recognized and approved for certain cancers. So it does the job very, very well, but it's limited because it uses a specific antibody for the pull down of the CTCs, whereas the other platforms that we're using, which are filter-based, size-based, don't have the rigor and the FDA approval of the Veridex system, but they have the advantage that they're not limited to a surface marker. So,
and genomics?
And genomics, well, my speciality, if you like, and the reason I've joined Carolyn Dives' group as her deputy and set up the genomics aspects there in the Paterson, is I've been working on single-cell profiling for 24 years now. So I started that off in the Ontario Cancer Institute in Toronto, working with a guy called Norman Isco, where what I was looking at there was hematopoietic stem cells. And we had to be able to work at the single-cell level because you simply don't have huge numbers of stem cells or their progeny. So 20 odd years ago I developed this methodology and have been using it since then. In the last 10 years I've been working in the biotechnology area as a research director and taken on some of that and made it into a commercial success in the form of a company. But I wanted to get back to the clinic because I can see that some of those technologies I've developed are now ripe and ready for actual implementation in the clinic, because it's all a matter of timing with these things. You can have a marvelous genomics technology, but if you don't have the bioinformatics or the computing to match it, you can't do anything with it. And I think everything is now converging in the area of oncology to a point where some of these very, very sophisticated technologies can start making a difference for patients in the clinic. And that's my...
Thanks, Ged. So we've done 1 year of the 5 years, and I think people seem to reckon we've done quite well in the first year of a project.
I would think so, yes. Yeah. I mean, again, it's the first time I've been in on this type of large European-funded grant, and I'm overall quite impressed by it. And I think there's a huge benefit in bringing together people who, as you say, they may know about each other, but it's a different matter knowing about somebody, but sitting down and working out practical plans, basically.
Ged, thanks very much. We'll speak to you in another year and see how the progress is.
Okay? That will be very good.